Thank you for giving me the opportunity to talk at this conference. It's a really great opportunity. Really appreciate it. So I'll just be talking a bit about some of the technology that we've been building at our company that helps predict protein ligand binding and could potentially be useful in a number of drug discovery contexts and potentially to CLCN4 NDD. So I'm sure you're all very familiar with AlphaFold and RosettaFold. If you're not, these were some pretty big sort of improvements in our ability to predict protein structure from the primary sequence of the protein. This has been a challenge that the scientific community has been trying to do for decades, and it was only in 2020 really where we really cracked it as a community. So we found a way to predict the 3D structure of a protein accurately from the sequence using deep learning. And that was done by DeepMind with AlphaFold, as well as David Baker with RosettaFold. And so David Baker is actually one of our co-founders. But then, so actually there's a small caveat, because the protein sequence itself does not uniquely determine the protein structure. The protein structure is actually affected by, for example, small molecules as well. So I've put an example over here. This is a protein called KRAS, which is very well known in the field of cancer. And when you look at the structure of KRAS without any small molecules bound, it looks like that in blue. And when you look at the structure of KRAS bound to a drug that treats KRAS-driven cancers called sotorasib, which is a drug made by Amgen, you can see that when the drug binds, it causes a change in the KRAS structure, revealing a pocket that is not normally seen in KRAS. And so these types of effects are very difficult to predict computationally, and it's really what we've been trying to focus on. And we've coined that term protein ligand co-folding, or protein drug co-folding. This is we believe that if we can solve this, that'll be a pretty big step forward in drug discovery, because you could then potentially find small molecule modulators of a number of different targets that are quite hard to do right now. So what we've been developing then is this sort of co-folding deep learning model called DragonFold that takes as input the protein sequence as well as a small molecule, and then it folds them together to produce what we call a virtual crystal structure, which is essentially a predicted 3D structure of the protein bound to the small molecule. And so this has been developed in collaboration with our co-founder, David Baker. So how do we do this? Well, it's a deep learning model, so it relies on a lot of training data. So the data source we use is the PDB, the Protein Data Bank. That now has over 200,000 structures that have been deposited by researchers since its start in the 70s. And as you can see here, over 25% contain small molecules. So we have over 50,000 protein ligand crystal structures from which to learn how ligands bind to proteins and how the ligands affect the structure of the proteins. And so we then train the model using a lot of GPUs. We have a supercomputer that we've built in collaboration with NVIDIA that is actually more compute than what was used to train AlphaFold. This is really a core part of the algorithm, training on a lot of data for a very long time using transformers. And then that was supposed to be a video of showing the folding process, but essentially it starts off by predicting all the atoms to be in the same place, and then it sort of updates the atoms of both the protein and small molecule sort of iteratively until it converges on the structure. This was to show that you can do this process for a single protein and small molecule, but then you could actually do it for you could do that process multiple times for different small molecules to screen, small molecule libraries against proteins. So we've then tested, how well this works in practice, experimentally, because we think prospective validation of the method is very important. So, for example, over here we took 3 different disease targets focused in cancer, but it could work for other disease targets for other therapeutic areas, and potentially rare diseases as well. And then we took a chemical library which contained around 2 million small molecules. And then we folded each molecule against each of the target proteins to generate a number of virtual crystal structures from which we selected the molecules that we thought would bind to the proteins. And then so for each target, as you can see, we selected only a few hundred compounds for experimental screening. And so in the first case, so the first protein is it's a PPI, a protein-protein interaction. So you're trying to disrupt the formation of 2 proteins sort of coming together. And we found, the best hit we found was single-digit micromolar, which is more potent than what you'd normally get from doing a high-throughput screen of a small molecule library. And we also found that it works for a polymerase. But the last row is saying that, so for the other protein-protein interaction, it didn't work. And we think that that's because that target had less training data or less relevant training data. And so we think the training data is very important for, trying to really, confidently know that you'll be successful on this target. So then we also wanted to have a look at how well it can predict selectivity. And so in other words, how well it can predict binding of a small molecule preferentially to one protein compared to another one. This is very important in the kinase field because so kinases are a class of proteins that all look very similar. And so if you have a small molecule that inhibits one kinase, it may well inhibit many other kinases, which is not ideal for a therapeutic. So what we did in this case was there was a kinase that we wanted to target, but there were 2 other kinases, off-target 1 and off-target 2, which were structurally very similar to the kinase we wanted to target. So we wanted to find hits, small molecule hits that only bind the kinase of interest. So what we did was, again, we did a co-folding of all of the molecules in the library against each of the kinases, all 3 of the kinases, to get the virtual crystal structures. And then we selected compounds that would only bind to the kinase we wanted, but avoid the other 2 kinases, which are very structurally similar. And in this case, we only screened 117 compounds. So, we're screening a very small, we're experimentally screening a very small number of compounds. And so, as you can see in the top row, what we see is that there's a competitor molecule which is, which recently entered the clinic, and that inhibits one of the off-target kinases as much as it inhibits the kinase that they want to target. And this might cause side effects in their clinical trial. And with the hits that we were able to come up with, they showed, in the first 2 cases, bottom left and bottom middle, around over 10-fold selectivity over the off-target kinases. And in the last case, the bottom right, it only inhibited the kinase of interest, and it completely avoided the other 2 off-target kinases, even though they're structurally very, very similar. So that could potentially be quite interesting. And so then, and I wanted to just sort of end here. So, how then could we potentially leverage a tool like DragonFold, to help with something like CLCN4 NDD? Well, the first thing that we could potentially do, and these are really just ideas. I'm not an expert in this disease, but we could potentially fold libraries of small molecules against targets that we think could modulate CLCN4 biology. And we could screen existing drugs, which would have certain advantages, but we could also screen novel drugs. And yeah, so that was sort of one of my thoughts, and I'm not the expert here, so I'd be very interested to hear all of your thoughts as well. And yeah, that's sort of basically what I had to say. I mean, I guess this is an overview of our company, but I'll probably sort of just skip that, as that's probably not that interesting. Yeah. Yeah.